the next step we have to do is we have to calculate statistics. And this is relatively simple. We could just say df dot mean and call the mean method, and that would very quickly calculate the mean values for all the columns. The problem is this only works if your entire data frame consists of numbers. But we can't do that because our data frame has both string values like the cell line names, the cancer types, and things like that, and it also has got numeric values. So we want to filter out just the numeric values first and then call the mean on those. So there's different ways of doing that. I'm gonna jump into the notebook to do that. Let's go through that. So I've already loaded the data set to avoid the wait. And yeah, let's have a look. So first of all, if we've imported pandas, we are ready to go with our data frame. That data frame is loaded. So if we just say df dot mean here, we will get an error because pandas doesn't like it because we have got non-numerical values in the data frame. So that we cannot do. So we need to find a way to get the first only the numeric column. So let's have a look at the column structure. So if we call columns, that's an attribute, that's the data on all, what are the names of the columns. And you can see here that our genes start with column 7. So 1, 2, 3, 4, 5, 6. The first 6 columns are string data, and then we start with the gene names, which are all the numerical, the gene dependency values. So this is a list, so we can filter it. We can say, for example, just start this list from 6, from the 7th entry, so from 0 to 6, and then the number from 0 to 5 are the string values, and then number 6 is the first numeric, this column here, and then go until the end. So we use the colon nomenclature to get to the end here. And that, if we do that, that will give us, now we have all the gene names here. And now we can say, for example, these are the gene columns. So let's call this gene columns. So this is a list now with all the gene columns. And now we can use this list to filter our data frame further. So we can say df gene call. So we provide the list of the column names that are just the genes, and that will give us our data. Whoa, this is large, obviously. So we don't want that. We want the mean for each of these columns. So these are all the this is actually the mean, not just of the breast cancer cell lines, but also the myeloid cancer cell lines. But let's get that mean out first. So let's just do mean. What happens then is that it calculates for each gene the mean value, and then you get a new data set that looks very different. So now it's turned around, and each gene gives is associated with the mean value across the cell lines that we're analyzing. And that's exactly what we want. That's where we want to get here. So this is incredibly fast. Pandas under the hood uses a C library that can analyze all these columns at the same time. And you can do this for millions and millions of columns at the same time, and it will be much, much faster than with other methods. And that's basically, there's other statistical analysis that that we can use. So we can ask for medians, for example, standard variation, minimum, maximum. So Pandas is really designed for this type of statistical queries. And there's quite a lot of practice again in a notebook. But right now we are almost there. So we have filtered the breast data. We have gotten to the numeric data and got the means for that. So now we have a list of all the genes and the mean dependency values for each gene for the individual breast cancer cell lines. So now we want to find which ones are the most essential genes in the breast cancer and then in the myeloid cancer panels.